Hello viewers, we are celebrating National Immunization Day in India. It is always celebrated on 16th of March each year. Immunization is a global health and development success story, saving millions of lives every year. Vaccines reduce risks of getting a disease by working with your body's natural defense to build protection. Immunization currently prevents 2 to 3 million deaths every year from diseases like diphtheria, tetanus, pertussis, influenza and measles. Now, the pioneers of vaccine development are Dr. Edward Jenner, who is remembered as the pioneer of the smallpox vaccination and also the father of immunology. Dr. Louis Pasteur, who is renowned for his discoveries of principle of vaccination, microbial fermentation and pasteurization. Dr. Maurice Hilleman, remembered as the father of modern vaccinology, as he developed nearly 40 vaccines. How vaccines actually prevent disease? Vaccines contain the same antigen or part of antigen or epitope that causes diseases. For example, measles vaccine contains measles virus, but the antigen in vaccines are either killed or weakened to the point that they do not cause disease. Now let's understand the different types of vaccines currently used. We can have a whole organism vaccine where the entire organism is being used. The organism is either killed or inactivated or attenuated or weakened. It can be a purified macromolecule as vaccine where a part of the organism or the pathogen, for example, it could be a toxin that is converted to toxoid or the capsular polysaccharide of the pathogen, or the recombinant microbial antigen or surface antigen is being used, that is a part purified macromolecule of the pathogen is used as a vaccine. It can be a recombinant vector vaccine, it could be a DNA vaccine, it could be a synthetic peptide vaccine, or it could be a multivalent subunit vaccine. Now, when we are talking about a whole organism vaccine, as already said, it could be an inactivated or a killed pathogen or a live, weakened, attenuated or avirulent bacterial cell or viral particle that is being used as a vaccine. Now, when we are talking about a whole organism vaccine, let us understand the live attenuated. So, in live attenuated, the pathogen has not been killed but rather weakened. So, the classical example I will take from each category, the bacterial cell in case of tuberculosis, the live attenuated, the BCG vaccine is actually the Bacillus calmette-guerin vaccine is actually a live attenuated tuberculosis bacillus that is used as a vaccine. Similarly, when we are talking about viral vaccine, in measles, the live attenuated virus is being used as a vaccine. Similar case in case of mumps also. Now, let me throw more light about the BCG immunity and tuberculosis. The BCG vaccine for percutaneous use is an attenuated live culture preparation of Bacillus of calmette and guerin BCG strain of Mycobacterium bovis. BCG was developed by growing M. bovis on a medium containing increasing concentration of bile. Tuberculosis is primarily an airborne communicable disease caused by bacterium Mycobacterium tuberculosis. Now, another live attenuated measles vaccine, here the virus is being attenuated. Measles is an acute illness caused by a virus of the paramyxovirus family. It is transmitted via droplets from nose, mouth or throat of infected persons. The virus infects the respiratory system, making it highly contagious. Globally, an estimated 450 people, mostly children, die every day from measles, despite the fact that we do have an effective and a safe vaccine, that too at a very low cost. Now, the first measles vaccine was tested in 1958 by Katz, an infectious disease specialist who was working with other researchers in Boston lab. He tested the first version of lab's vaccine on mentally retarded and disabled children at a school outside Boston. Each of the 11 vaccinated children developed measles antibodies, but 9 also developed a mild rash. The vaccine didn't cause full-blown measles, but it did cause the symptoms. So, the researchers finally realized the virus used for the vaccine has to be weakened even more. So, I am just taking this example because this is how the vaccine development actually took place. Later, attenuated measles vaccine developed was developed by Maurice Hillman and his colleagues, and this was attenuated by passing the measles virus, which was used by John Edmer, and it was passed through different cell types 80 times. So this is how they attenuated the measles virus and used it later as a successful measles vaccine. Now, another example which I took for the attenuated virus vaccine is a mumps vaccine. Mumps, we all know, is a contagious disease caused by a virus that leads to a painful swelling of parotid salivary glands. Mumps mostly occur in children of the age 2 to 12 who have not been vaccinated. Why did I took up this example? Was because it had a very interesting story. Now, mumps symptoms are basically fever, headache, painful swollen glands in face, neck and jaw. Now, what happened? Hillman was the first one who isolated the mumps virus in 1963 from his daughter Gerald Lynn. During her illness, he actually took out the virus from her. Now, what he did was, Hillman's 5-year-old daughter was feeling ill. He woke up in the night. He believed that she probably had mumps, as she had all the typical symptoms of swollen glands, fever. Now, though Hillman was worried about his daughter, yet sensing an opportunity, he drove to his Merck office to pick up the cotton swabs in a vial of broth. Once he was back home, he swabbed the Geraldine's throat, placed the swab in the broth and kept it in the freezer, and later he used it as a vaccine for his younger daughter. In this slide, you will see that he actually inoculated his younger daughter from the attenuated vaccine or from the attenuated virus which he collected from his elder daughter. Now, another way where whole organism vaccines are used is by inactivating or killing the pathogen. The typical example I have taken is anthrax, which is where a whole organism is also used as vaccine, besides various other illnesses like cholera, pertussis, plague, etc., where vaccine is actually the pathogen which has been killed. In case of viral pathogens, the vaccine developed against hepatitis A, influenza or the SARC virus is actually a whole organism killed pathogen vaccines. Now, I have typically taken anthrax because anthrax is a very important biological warfare material. You can see these were the envelopes which were sent to senators and mailmen, which was specifically sent by the terrorist groups after 9/11, and it contained the anthrax spores. Now, anthrax infection can take place by 3 ways. The causative organism is Bacillus anthracis, it is a bacterium. The inhalation of this bacterium causes severe flu-like illness with high mortality. Unless diagnosed and treated immediately with antibodies like penicillin, doxycycline or ciprofloxacin. Now, the cutaneous exposure can result in skin lesions with characteristic black deep edge scar. Cutaneous anthrax has almost 20% mortality, while GI exposure, that is gastrointestinal exposure, can result in ulcers in ileum or cecum, and in almost all cases this is the most fatal one, leading to 100% mortality in such cases. Now, how anthrax vaccine actually is made? Now, this is slightly about the toxin vaccines which I had mentioned previously also. The anthrax vaccine is made by taking a strain of bacterium Bacillus anthracis and growing it in laboratory. The bacterium actually release certain harmful proteins. Of course, the attenuated or basically killed bacterium is also used as vaccine, but there is another way of using the toxin which is released by the Bacillus anthracis. This toxin is taken up from the surrounding broth once the bacterium is growing in the broth material. These toxins are responsible for disease in those infected with anthrax bacteria. The toxins are then inactivated using formaldehyde so that they are no longer causing diseases. Such inactivated toxins are called toxoids. Then this toxoid is taken out and it is used as a vaccine, just like we use the pertussis vaccine or diphtheria vaccine or a tetanus vaccine, which is an acellular vaccine. There is no cell involved. It is a toxoid, that is the inactivated toxin, which is used as a vaccine to take care of people. Now, Jonas Salk and the polio vaccine. It is very important to talk about the polio vaccine. Salk began the early polio vaccine trial in 1962. What he did was, he began with the trial of the Salk vaccine. He used the Salk virus and killed it or attenuated it and started injecting it into the people. For the first time he used this vaccine against for his family only. As you can see in the slide, it's his wife and his kid who's been right now injected by the Salk vaccine. Now, unfortunately, when the result was announced by University of Michigan, the government agreed to use this vaccine. However, it was found later in 1955 that this vaccine led to, it was insufficiently killed and it led to infection in almost 200 children. Many of them got paralyzed and several died. So, by 1955, this polio vaccination was suspended. The full review actually concluded that 11 people died from the vaccine and 100 got paralyzed. This resulted in failure to completely eradicate polio. Later, another vaccine which was very important came up was called the MMR vaccine. This vaccine give protection against mumps, measles, rubella. Why this vaccine example has been taken? Because this was the first vaccine which was a combination vaccine. That is, it was a combination of 3 vaccines against 3 different infectious agents. Now, MMR combination vaccine debuted in 1971. It was licensed by Merck and it was a very successful vaccine. Now, later purified macromolecules as vaccines were also used. As already said, I had taken the example of using toxoid macromolecule as vaccine when I was talking about anthrax also. For diphtheria, tetanus also, the exotoxin. What is an exotoxin? It is a toxin which is secreted by the bacteria. That is being inactivated, a toxoid is formed and then that is used as a vaccine. Or the capsular polysaccharide, the outer component of the pathogen is used. For example, in case of Haemophilus influenza type B or Neisseria meningitis. Or a surface antigen protein is used, for example, in case of hepatitis B. Now, toxoids are manufactured from bacterial toxins. Some bacterial pathogens, like as I told you in case of diphtheria and tetanus, produce exotoxins. That actually is the causative agent of the disease. They lead to symptoms that result from infection. Tetanus vaccine, for example, can be made by purifying the bacterial exotoxin and then inactivating the toxin with formaldehyde to form a toxoid. This is how the tetanus vaccine is formed. Exotoxins are proteins produced inside the pathogenic bacteria, mostly gram-positive bacteria, and are the part of the growth and metabolism of the bacterium. These exotoxins are secreted or released into the surrounding medium following the lysis of the bacteria and then further weakened and used as a vaccine. Now, bacterial polysaccharide capsule are also used as vaccines. Like for example, in case of influenza, Haemophilus influenzae type B, which is a major cause of bacterial meningitis in children less than 5 years of age, the outer bacterial polysaccharide capsule is being used as a vaccine. Vaccine for Hib consists of type B capsular polysaccharide. It is covalently linked to a protein carrier, tetanus toxoid. It has been found if you link it to tetanus toxoid, the efficacy of the vaccine increases much more times. It activates T helper cells in the person who has received this vaccine, enabling class switching from IgM to IgG, which we know IgG is produced at a humongous level and can also lead to memory B cell formation. Now, proteins from pathogens are also produced by recombinant techniques. The gene encoding any immunogenic protein can be cloned and then expressed in bacterial yeast or mammalian cells by rDNA technology. A number of genes encoding surface antigen from viral, bacterial, protozoal pathogen have been successfully cloned into bacterial, yeast, insect or mammalian expression system, and the expressed antigen is used for vaccine development. The first such recombinant antigen vaccine which has been approved for human use is hepatitis B vaccine. Hepatitis B is a viral infection that attacks the liver and can cause both acute and chronic diseases. The virus is transmitted through contact with the blood or other body fluids of an infected person. More than 7,80,000 people die every year due to consequences of hepatitis B. This vaccine, hepatitis B vaccine, is 95% effective in preventing infection and its chronic consequences. Now, Recombivax HB is the vaccine which is produced against hepatitis B virus. As you can see, this is the strategy by which the RNA portion of RNA from hepatitis B is taken. It is cloned into a bacterial plasmid DNA. The recombinant DNA is then put into a yeast expression system. Once it is into the yeast expression system, it is put into fermentation tank and yeast undergo replication, and many yeast cells are produced, and the recombinant protein is then expressed and it is taken up, and then finally a formulation is used. Formulation is made which is further marketed as a vaccine. There is another type of vaccine called multivalent subunit vaccine. Now, what happens in this case? Incorporation of multiple antigens from the same microbe. You must understand, when we are using many epitopes, it will lead to production of a polyclonal sera. That means different antibodies against different epitopes will be produced and will be much better. It will have much better efficacy to combat any pathogen in such scenario. So, such multivalent subunit vaccine have incorporation of multiple antigen or epitopes from the same microbe into a vaccine through construction of SMAA, which is solid matrix antibody antigen complex. How it is made? Around a solid matrix, we attach different monoclonal antibodies which are specific to different epitopes of a particular pathogen. So, once we have kind of made an opsonin on a solid matrix by these monoclonal antibodies, different epitopes are conjugated to these antibodies, and finally an SMA, that is solid matrix antibody antigen complex, is formed, which when injected into a recipient will produce a better efficacy in combating a particular pathogen. There is another vaccine type called DNA vaccines, in which in such kind of vaccines, portions of the pathogenic DNA is directly either combined with a plasmid DNA or a carrier DNA and is directly injected into human cells, which make proteins that elicit an immune response to the pathogen. The encoded protein of DNA vaccines is finally expressed in host in its natural form. DNA vaccine has an advantage. It leads to induction of both humoral as well as cell mediated immunity. These vaccines also cause prolonged expression of the antigen, which generates a significant immunological memory. Now, there is another new vaccine, that is cancer vaccine. In this case, tumor cells are excised or taken from the tumor cell line from the person who is having cancer. From that same person, we isolate the dendritic cells. What they do is, the tumor cells are inactivated, and once they are mixed with dendritic cell from the patient, these are then injected back into the patient as immunogen. Now, what exactly happens is, once the dendritic cells picks up some DNA or part of the cancer cell, this new dendritic cell will act as an APC, that is an antigen presenting cell, and will present the tumor portions to the T cells of the same individual, who will produce a heightened immune response against the cancer pathogen. In these two examples, antigens are introduced directly into the dendritic cells or generated in situ using tumor RNA. The cells then produce tumor antigen and hence direct the T cells to seek out and destroy the cancer cells. Now, as you can see, a lot of work has been done. This slide actually shows how the disease occurred, and finally the disease was, the vaccine developed, and finally the disease was contained. Now, if I primarily want you to see the Ebola, Ebola has taken the third from above. The Ebola outbreak has occurred twice, 2014, 2016, 2018. Vaccine development took place way back in 2019. The disease is yet to be controlled. It keeps coming up now and then. Now, as I just told you, it came up in 2014, 2016, 2018. There are 4 stages of Ebola. By 4th stage, I mean, which maybe on 12th day or the 13th day, maybe on the 12th day, the person already reaches coma. So, it affects right from 7th day to almost the 12th day, the person is comatose. It's a very deadly infection. The Ebola virus disease, formerly called Ebola hemorrhagic fever, is a severe fatal illness in human. The virus is transmitted to human from wild animals, and it spreads in the human population through human to human transmission through contact with bodily fluids like sweat, tear, urine, semen, etc., or through bedsheet, clothing, needles and corpses also. An outbreak in 3 Western African countries led from 2014 to 2016 resulted in more than 28,000 cases of EVD and around 11,000 deaths that were caused by Zaire Ebola virus. In December 2019, US FDA, that is Food Drug Administration, announced the approval of Ervebo, which is the first FDA approved vaccine for prevention of Ebola virus. Now, there is another virus, that is the Zika virus, which is actually because of mosquito bites, which keeps coming up again and again. There was an outbreak in 2015. It is a disease caused by a virus that is transmitted primarily by Aedes mosquito, which bite during day. Zika virus infection during pregnancy can cause infants to be born with microcephaly and other complications. It can also lead to many complications in pregnancy and preterm birth or miscarriages. Currently, there is no treatment or vaccine for Zika virus. Symptoms are generally mild and they generally last 2 to 7 days. In India, mostly Zika outbreak has taken place in the northwestern state of Rajasthan. Let's talk about COVID-19 now. Coronavirus disease 2019 is a contagious disease caused by severe acute respiratory syndrome coronavirus, that is SARS-CoV-2. The first case was identified in Wuhan, China in December 2019. The disease has since spread worldwide, leading to an ongoing pandemic. Symptoms of COVID-19 are fever, cough, fatigue, breathing difficulties, loss of smell, taste. Of those people who develop noticeable symptoms, 81% develop mild to moderate symptoms, while 14% develop severe symptoms and 5% are critical. The virus that causes COVID-19 spreads mainly from one infected person who is in close proximity to another person. Now, since its emergence, the SARS-CoV-2 virus has continued to evolve, and WHO has designated 5 variants as SARS-CoV-2 variants of concern, that is VOCs, to date, namely alpha, beta, gamma, delta and omicron, due to their impact on transmission, disease severity or capacity for immune escape. As per WHO, while omicron variant is spreading rapidly across world, today itself it is rampant in China, Hong Kong, New Zealand, Germany. The evolution of SARS-CoV-2 is definitely expected to continue, and omicron is unlikely to be the last VOC, so we need to be very, very careful. Now, these are few of the vaccines that came up. For example, the Pfizer vaccine, which was mRNA based, the Moderna vaccine, which was again mRNA based vaccine, CoronaVac by Sinovac, the Covishield, which uses adenovirus as a vector, which was developed by AstraZeneca, Sputnik, the Russian vaccine by Gamaleya Research Institute, the Covaxin, which was developed by Bharat Biotech ICMR, which is again an inactivated vaccine. As of 12th January 2022, the following vaccines have obtained emergency use listing, that is EUL: the Pfizer BioNTech Comirnaty vaccine, Covishield, the Janssen Johnson & Johnson vaccine, the Moderna COVID-19, Sinopharma COVID-19 and Sinovac Coronavac vaccine. They have all been given the EUL, that is emergency use listing. The Bharat Biotech Covaxin also received it on 3rd November 2021, Covovax on 17 December 2021 and Nuvozovid on 20th December 2021. Two vaccines that are currently being used in India are the Oxford AstraZeneca vaccine, which is COVID Shield, and another which is Covaxin, which is actually using an inactivated virus by Bharat Biotech along with ICMR, that is Indian Council of Medical Research. This is the vaccine R&D map of India. There are around 26 vaccine R&D institutes in India that are actively engaged in research and development of vaccines. Now, the development of next generation of vaccine will be more challenging, as many of the organisms which are targeted have complex structure and life cycle. One such is the malarial parasite, but thankfully WHO has announced the first malaria vaccine in October 2021. Now, another one, we have to outwit the human immune response, like for example in case of HIV, influenza virus, even for SARS-CoV-2. New strategies are constantly being explored to develop new vaccines for these diseases. As Nelson Mandela said, life or death for a young child too often depends on whether he is born in a country where vaccines are available or not. Hence, we must understand that vaccines save lives. Let us protect our little ones by timely vaccination. Again, भूल ना जाना, टीकाकरण जरूर करवाना. Thank you very much.